Hello and welcome to our today's lecture of NPTEL course Introduction to Mechanobiology. So, in the last two lectures I had discussed actin dynamics in cell migration, okay. And while discussing how cells migrate, we had found out that you have a molecular clutch mechanism which operates at the leading edge. Okay. So, what you have three situations. So, what I am drawing here, these are your integrins, okay, and let us say this is your actin filament, okay, and you have monomers which are getting added to the actin filament. So, when the clutch is disengaged, so this is a situation where the clutch is disengaged. Okay. So, when the clutch is disengaged, the polymerization, so let us say this is my leading edge position, okay. Okay. No matter how much the filaments form, they will just keep getting pushed backwards. Okay. So, this is your new filament which has gotten added to the existing filament, okay. And this is called your so this overall you have this backflow, and partly this is mediated by your myosin molecule which pulls on it, okay. So, you will have retrograde flow but no forward movement. In the other case, okay, when you have clutch is engaged, okay, you have various adapter proteins which link you with the filament. Okay. So, let us say this was the earlier position. In disengaged position, this was the leading edge position. So, when your clutch is engaged, so in this case your clutch is engaged, okay. So, your retrograde, so even when myosin motors are pulling on it, so what you will have is tractions on the substrate, and plus you will have protrusion. Okay. So, this is the distance by which the cell moved forward. So, this is the principle of a molecular clutch. Okay. Now, so imagine a situation, okay, you have, so this is your cell membrane, okay, this is your ECM, you have your integrin molecules here, integrins here, okay, and then various other proteins, right. So, I am just drawing these layers, and eventually you have your actin filament. Okay. So, you know that as a function of z, if this is my z height, okay, you have your integrins, then you have molecules like paxillin and FAC, on top of which you have vinculin and tailin. So, tailin exhibits a direct orientation, and on top you have alpha actinin and actin. Okay. So, if actin is so you have measured retrograde flow, right. So, you have measured retrograde flow. So, so at the bottom you have your ECM. Okay. So, flow at this point has to be 0. So, if I were to characterize the speed, what you should have here is 0, there is no slip condition at the base, and at the maximum, let us say at this blue point you have a certain speed here. Okay. So, let us say this is my axis. This would mean that all these intermediate proteins will have some speed, okay, some sliding speed which scales with height, and this is what has been found. Okay. If you track the motion of different proteins at focal adhesions, okay, you have integrin, you have FAC/paxillin, you have tailin, you have alpha actinin and actin. Okay. So, if you track the average speed of these molecules, okay, what you find is this guy has the high speed and then you have reducing speed. So, paxillin and vinculin are paxillin and FAC are way less than tailin or vinculin, okay, and lowest very close to 0 is integrins. So, this shows you that there is a hierarchy, okay, and this. So, in order to achieve this kind of speed, you must have transient connectivity. Okay. You have to have transient connectivity of focal adhesion proteins for this motion to be transmitted from the top to the base, where speed is 0 at the base, and as you go up you have different speeds. Okay. So, these two papers that we discussed would be our next reading assignments. Okay. First one, you have Ponti et al. Science 2004 paper. So, this is about the actin dynamics, and the second paper Hu et al. Also a science paper in 2007. This discusses about this relative sliding of different focal adhesion proteins. Okay. So, this concludes our discussion of mesenchymal migration. Now, compared to mesenchymal migration, so you have mesenchymal or adhesion dependent or adherent. So, this kind of migration is exhibited by epithelial cells, okay, fibroblasts, okay, cancer cells. So, there are various types of cells which exhibit this migration, but you also have another mode of migration which is called amoeboidal migration. Okay. So, today we will start discussing amoeboidal migration. So, what is amoeboidal motility? So, if you look at dendritic cells, okay, if I were to outline the shape of a dendritic cell, it will be more like this. Okay. So, what you see is, as opposed to the well defined shape of these adherent cells, these dendritic cells, they do not have a well defined shape. You have multiple different protrusions which are getting, which are evolving in space and time. Okay. And amoeboidal motility or amoeboidal migration is characterized. Okay. So, amoeboidal migration is characterized by frequent changes in shape. Of cell. So, amoeboidal migration is exhibited. So, if I were to measure the average speed of fibroblasts, okay, or epithelial cells, it be order, let us say 20 to 30 microns per hour. Okay. So, in comparison to this adhesion dependent type of migration, the speed of amoeboidal cells can be 100 times faster. Okay. So, the question that I want to ask is motility or is amoeboidal motility adhesion dependent or independent? Okay. So, the idea being if something is adherent, you are protruding and then you are breaking your rear adhesions. So, that should limit how fast a cell can move. In contrast, amoeboidal migration can happen very fast if there is no adhesion. So, is there any justification for saying that amoeboidal motility must exhibit adhesion independent migration? Okay. So, to answer that question, what I will do is I will first review. Okay, I will first review some aspects of amoeboidal migration. Okay. So, AM is short fall for amoeboidal migration. Okay. So, generally dendritic cells are much smaller, okay, than cells like fibroblast, than mesenchymal cells. Okay. So, there are, in case of amoeboid cell motility, there are, even on a 2D plane you can have 3 different types of motility. Okay. So, let me draw again. Okay. You have these adhesions, okay, and you have the nucleus. What I will also draw, okay, these protrusions I can draw in blue. Okay. So, you have 3 things. Okay. Blue is protrusion, okay, actin dependent. Okay. A is adhesion. Okay. A is adhesion, and what I have depicted here in red, okay, is C or contractility. Okay. So, 2D protrusive, a 2D protrusive can be drawn in the following way. Okay. You have 3 different aspects of it: protrusion, adhesion and contraction. Okay. When all 3 are dominant, this is nothing but almost equivalent to mesenchymal migration. Okay. So, cell protrudes, stabilizes the protrusion by adhering, and then the cell contracts. Okay. So, you have all 3 of these parts playing major roles, but you can have. So, remember that I made this statement, dendritic cells are much smaller than mesenchymal cells. Okay. So, what has been observed is these cells, okay, they can migrate just as well, okay, if you have small c. Okay. So, in this case I have drawn C to be very small, suggesting your contractility is very low. Okay. So, even with less contraction, if they combine protrusion and adhesion, the cell is able to migrate. And how is this possible? So, it is possible only because the cell is very small. Imagine that you take a balloon, okay, a balloon, and you poke from inside the balloon, you poke with a pin. Okay. Then what you will have is the balloon will slowly take this kind of a shape. So, what you are doing is the balloon membrane, you have a tension, a membrane tension which goes up. Okay. As a consequence, it is this tension. So, if you are adherent, then the membrane tension itself can lead to pulling the cell forward. Okay. So, this is an alternate mode where protrusion and adhesion are dominant and contractility is very low. Interestingly, for these cells there exists a third mode of migration. Okay. Nucleus. Okay. So, what it does? So, this is called a bleb based migration. Okay. So, this is called bleb based migration, in which your protrusion is very small, adhesion is large and contractility is large. Okay. So, the cell actually, so it is like you are exerting these contractile forces from the back, which leads to flow of the cytoplasm, leading to rupture of the membrane and creation of a bleb. So, this is called a bleb. Okay. So, and you can keep on doing this repeatedly, that you have a contraction, the cell moves forward, adhesions are broken and so on and so forth. Okay. So, this kind of bleb based migration has also been observed in dictostelium. Okay. So, you have these three cases. Now, this is on 2D surfaces. Okay. In 3D context, you can have various different situations. Okay. You can have a situation. Okay. So, what I am drawing around the cell, so this is 3D, that means within interstitial matrices. You can have this kind of migration where adhesion is small, okay, contractility and protrusion are large. Okay, this is one type of migration. Okay. You can have another piece of migration where your adhesion and contractility are small and protrusion is large. Okay. So, this is 3D protrusive. And the third mode is 3D contractile. Okay. In 3D contractile, your adhesion and protrusion are small and contractility is large. Okay. So, 3D protrusive can be sustained in matrices where pore size is comparable or greater than nucleus size. Okay. This is because the nucleus, so the nucleus is the stiffest compartment, stiffest and largest organelle. Okay. So, the nucleus being the stiffest and the largest organelle, for 3D contractile, okay, this is where matrices where pore size is lesser than nuclear dimensions. Okay. So, here you have protrusion playing a bigger role, here you have contractility playing a bigger role. Okay. So, in 3D contractile, so if I were to draw 3D protrusive, and you have, so the way I have drawn it, so this is 3D protrusive, and in 3D contractile, okay. So, in this case the nucleus, the 3D contractile, so this is 3D contractile. So, in the case of 3D contractile, the nucleus has to be deformed. So, again in 3D contractile you have a bleb based migration. Okay. So, this is just a brief overview of all the different types of amoeboidal motility you might have. So, now we will initiate discussion about one particular paper. Okay. So, the name of the paper is Adaptive Force Transmission in Amoeboidal Cell Migration. Okay. So, Nature Cell Biology paper 2010 paper. Okay. So, the broad goal here of this paper is to understand how do dendritic cells migrate in response to adhesive and non-adhesive substrates. Okay. The authors worked with dendritic cells. Okay. You have dendritic cells, these were transfected, okay. These were transfected with LifeAct GFP. So, LifeAct, okay, LifeAct is an entity which binds to filaments. So, as a consequence you can track the dynamics. Now, binds to filaments, but without perturbing actin dynamics. Okay. So, basically the first question the authors asked whether they would observe retrograde flow in these cells. Okay. So, to answer this question, the cells were plated on polylysine coated surfaces, okay, and what they observed. So, now how do you quantify retrograde flow using LifeAct GFP? Okay. So, this is what is done. Okay. If you have a cell, okay, so when you transfect with LifeAct GFP, you will see the entire thing fluoresce. Okay. So, now let us take a portion, a small window of the cell where the cell is actually protruding. Okay. So, what you can do, you can generate what is called a chymograph. Okay. Chymograph, and what the chymograph does is along a very small length of it, okay, along a very small length of the membrane, the cell tracks how the fluorescence signal and the membrane front evolve. Okay. So, as a consequence, if you look at the same line, okay, if you look at the same line and see how the fluorescence will evolve, what you can get. Okay. So, you have the following thing, you have a time axis here and you have a length axis here. Okay. So, let us say at time t equal to 0, you had some, let us say, distribution, okay, you had some distribution of the fluorescence signal. Okay. Let us say I draw it like this. Okay. So, this is one strip. Okay. The next time, okay, in the next time instant, I would plot it again. Okay. Similarly, the next time instant I would plot it again. So, what I will get is these streaks I will get, and if I connect these streaks, so this is how a given point in the membrane evolved as a function of time, and what you would see is a line. Okay. So, this is time and this is length. Okay. So, this length is taken from this direction backward. Okay. So, this point is the leading edge, okay, and this is for the width, it is some point which is inside the cell. Okay. So, by connecting these dots, I will get these lines, okay, and what you find is, so because this is the leading edge and this is the trailing edge, this is some point inside the cell, a slope this would give us retrograde flow. So, you have a given retrograde flow. So, you can measure, you can do multiple measurements and you can get some value of retrograde flow. Okay. So, in terms of microns per minute or microns per hour. So, under control treated conditions, cells, the authors observed some retrograde flow. When they perturbed actin polymerization, okay, with this drug called latranculin. Okay. So, what latranculin does is it sequesters G actin. Okay. As a consequence, your polymerization is stopped, and what they found is, okay, in latranculin treated cells you have the same amount of retrograde flow. Okay. So, you have two conditions in which the retrograde flow does not change when you perturb the polymerization. So, what does it suggest? It suggests that probably it is again the myosin motors behind the lamellipodia which is pulling the entire network backwards. Okay. Now, if this was true, then if you do experiments with lat plus blebistatin, okay, blebistatin is the myosin inhibitor, what they observed was that in lat plus blebistatin this was completely eradicated, your retrograde flow is completely eradicated. So, this suggests that myosin contributes to retrograde flow. So, I will stop here for today and we will continue from here in the next class. Thank you for your attention.